Hello all, welcome to this session for the board exam revision in the subject of biology. Today I will be discussing some important concepts of the chapters strategies for the enhancement of food production, microbes in human welfare, biotechnology principles and processes, biotechnology and its applications. We'll start with the strategies for enhancement of food production. In this chapter, food is considered both animal and plant-based foods. So to enhance the quality of food, if we consider animal-based food, what are the products of animals which you consider as food? One is milk, in the animals that give milk, and meat. Milk and meat, the quality of milk and meat. So in this process, you need to increase the quality of the animals that are considered as food, and also you need to increase the conditions in which these animals are reared. So animal husbandry is the branch of science that deals with the rearing of animals and taking care of them. In animal husbandry, you have to know a bit about dairy farm management, poultry management, apiculture. Apiculture is beekeeping, and pisciculture. Pisciculture is fisheries. In dairy farm management, whether it is dairy farm management, poultry management, in these both, the conditions for the maintenance of these animals are kind of similar. You have to take care of the selection of the animals, the right kind of animals, the right breeds you have to select, housing of these animals, nourishment, the supply of food and water, the conditions which should be hygienic. There should be care taken for their health. So if you take care of all these processes, if you take care of all these things with the management of these animals, the output of food in these animals will be better. The output of dairy is milk and meat. Both in poultry management, there are two types of birds that are reared. One for meat, which are called broilers, and the one for eggs, which are called layers. Remember that this can be one more questions. Broilers are reared only for the purpose of meat. Layers are reared for the purpose of eggs. The conditions, the principles of poultry management are similar to that of the management of dairy. In apiculture, in beekeeping, you can keep the beehives or the bee boxes in any place which has access to a floral field, whether it's a Apis cerana indica. Pisciculture, there are two types. One is freshwater fisheries and the other one is saline water. Aquaculture is freshwater and pisciculture is saline water. In fisheries, both. So in fisheries, you can do it in natural waters or in fish ponds. Animal husbandry, and now we look at some of the principles of improvement of crops, plant crops. For improving the crops in plants, and before we go into plants, a little bit of strategies that will help in improving the quality of these breeds. For improving the quality of animals that are reared for food, you do crossing with superior animals, crossing between superior animals. Cross is the mating between animals. There are two types of cross that you can do. That is inbreeding and outbreeding. Inbreeding, breed is a group of animals which have a common ancestry, common line, and they are very similar in their characteristics. Like if you look at dogs, there are multiple breeds. Retrievers are a breed, Alsatian is a breed, different breeds. They have similar characteristics and they have a similar descent. The closer if they are in the same line, you call them inbred population, inbreeding. If you want to cross, inbreeding, as we discussed in genetics, inbreeding will lead to improvement or increase in the number of characteristics that are present in those lines. The stronger characteristics will get expressed more, and the recessive characteristics, if there are some defective genes in the recessive traits, will get exposed and they can be eliminated by selection. So inbreeding will lead to creation of pure lines. But one problem with inbreeding is with repeated breeding within the same lines, there is depression of reproductive vigor and vitality. This is called inbreeding depression. Inbreeding depression can be overcome by crossing this inbred animals with one generation. Crossing is sufficient. Once you cross them with some other superior animal of the same breed, the inbreeding depression can be overcome. Outbreeding has different varieties. Outbreeding or outcross is between the different breeds, between different lines of the same breed is outcross. Cross breeding is bringing about mating between animals belonging to different breeds of the same species. The third type of outcross is interspecific hybridization. Interspecific hybridization is when you mate two animals belonging to different species, like the mating between a male donkey and a female horse gives rise to mule. Interspecific hybridization often does not lead to viable progenies. Wherever they are viable, they are often sterile. So these are the varieties of crosses that you have: inbreeding, outbreeding. In inbreeding, you have the one thing you have to remember is inbreeding depression. Inbreeding depression can be overcome by a crossbreed or a cross within the same breed. In outbreeding, remember crossbreeding, outcross, that is between individuals of the same breed, not in the same line. Crossbreeding between different breeds of the same species. Interspecific hybridization between individuals belonging to different species. This much of animals you have to remember. Two more strategies to improve the quality of animals that you get in this, improve the quality of animals is: one, artificial insemination, and the other one is multiple ovulation embryo transfer, MOET. These two can be questions. Artificial insemination is, see, if we consider cattle, you have a superior cow, you have a superior bull and a superior cow. The superior bull cannot be naturally mated with multiple cows. So you can collect the semen from the superior cow, superior bull, and inseminate through an inseminator different cows. The advantage of artificial insemination is the semen from a bull can be simultaneously at the same time inseminated into multiple cows, bringing about an increase in the number of progenies without involving the bull for mating. And this allows the superior bull to contribute semen or sperms to cows at different locations geographically. So the problem of geographical distance is overcome by artificial insemination. Multiple ovulation embryo transfer. See, artificial insemination has its limitations also. That is, it may not be successful. The success rate of artificial insemination is not very high. So what is done? The cow, a superior cow is selected, a superior bull is selected. The superior bull semen is the cow is prepared by stimulation with hormones like FSH to bring about multiple ovulation. The superior cow is producing multiple ovules now, multiple ova. The semen from the bull, superior bull, is inseminated into this cow. There is stimulation of the production of multiple embryos. Before these embryos get implanted into the uterus of the cow, the embryos are non-surgically extracted from the uterus of this superior cow and will be put into the uterus of several cows which have been prepared for implantation. There are two things happening here. One, collect the semen from the superior bull, inseminate it into a superior cow which has been stimulated for multiple ovulation. Second, parallelly when this is happening, the different cows are being prepared for implantation. Embryos are taken from the superior cow, put into the prepared cows. Now these cows which receive the embryos, they were not the ones which belonging to that superior quality, but they are harboring a superior progeny. This increases the number of superior progeny produced. Plant breeding techniques. The quality of improvement in plants can be by selection, hybridization, mutation breeding, and the fourth one is plant tissue culture. Selection is simple. That is the oldest method. You look at the plants for the favorable traits, select the ones which has the favorable traits, reject the other ones, keep breeding these things repeatedly, and over a period of time you have an entire species. You have an entire embryos or the seeds which produce the plants with the desirable characteristics. Selection. Hybridization is grafting is also a mode of hybridization where you bring in two plants, where you cross two plants with desirable qualities and expect the qualities to amplify. Just because you cross two plants with desirable qualities doesn't mean that the quality that you want will be expressed. It's a matter of chance. Again, reproduction is a matter of chance. The expression of characteristics is a matter of chance. Even there, selection has to be applied. Here also, it's a matter of chance. The fourth one, plant tissue culture. plant tissue culture or micropropagation. This is the latest technique which enables the qualitative plants, the plants with superior quality, to be propagated in large numbers very quickly. One characteristic of plant cells is exploited in tissue culture, that is the totipotency of cells. Totipotency means any living plant cell with nucleus is capable of producing the entire plant if stimulated in the right way. None of our cells, none of the human cells are totipotent. We have pluripotent cells, like the stem cells in the bone marrow are pluripotent. Pluripotent cells are the ones which will give rise to different types of cells, not a single type of cell as they grow. Totipotent cells gives rise to the entire organism. So plant cells are totipotent if they are nucleated and living. Take these plant cells, stimulate. I'll just cut it short. You take any tissue from any living tissue from the plant body, put it in a nutrient medium enriched with nutrients and hormones like auxins, gibberellins, and cytokinins in proper proportions. Let's not worry about the hormones, what hormones will give rise to what development. Hormones in the nutrient medium. Allow that tissue to grow. It will form into, it will transform. Plants will have meristematic cells which will differentiate to form permanent cells. Now, when stimulated in this medium, which is used for tissue culture, the differentiated cells will de-differentiate. That is, the meristematic cells had become permanent cells, that is differentiation. Permanent cells will go back to becoming meristematic cells, that is de-differentiation. This leads to the formation of a mass of undifferentiated cells called callus. You take this callus, you can make callus culture. You take the callus, transfer it into another culture medium. You can continue to build callus cultures. Or when you want the plants to grow, you stimulate the callus in a given medium by the proper hormones, and you will have the shoot and root differentiation happening. So with plant tissue culture, you have to remember the differentiated cells will de-differentiate, and when stimulated appropriately will redifferentiate to produce an entire plant. Advantage: you can select any plant, you can have the clones of these plants in large numbers very quickly. This is the greatest advantage of plant tissue culture. I think this chapter, strategies for the enhancement of food production, we can stop it here. I have discussed most of the important concepts here in this chapter. Microbes in human welfare. Microbes are used extensively for variety of our needs. The entire day of ours is dependent on microbes. It's useful in food, the fermentation of foods that we eat, the conversion of milk to curds, the formation of cheese, production of alcohol, industrial uses, agricultural uses, use of microorganisms for medicinal purposes, use of microorganisms in sewage treatment. A range of usage for microorganisms in various aspects of human life. One small concept in this chapter, this chapter requires you to remember the names of several microorganisms and what is their utility. I'll not go into that because it will be a listing of certain things. It will be a listing of microorganisms and their benefits. Rather, I would run through this and get into biotechnology, which has some very important concepts there. In sewage treatment, there are two stages of sewage treatment: primary treatment and secondary treatment. In primary treatment, you remove all the sediments by multiple stages of filtration and sedimentation. Then you transfer this, you take the sewage, the left out, the water for secondary treatment. In secondary treatment, you aerate the remnants that are remaining, which will increase the level of oxygen in the water to be treated. That makes the microorganisms present in that utilize the organic matter. Here you need to remember one concept: biological oxygen demand. Biological oxygen demand is the amount of oxygen required to completely oxidize a unit of organic matter present in a medium. So if more organic matter is present in a medium, there is more demand of oxygen to oxidize it completely. It also, in another way, indicates the amount of oxygen being consumed by microorganisms in a given medium. If there is more organic matter, there will be more microorganisms eating away on that. So it's an indicator of pollution. If biological oxygen demand is high, it means that there is higher organic matter in that medium, and there is increased microbial growth in the medium. So remember biological oxygen demand. And subsequently, there the slurry, aeration slurry is allowed to settle down. That is the cluster of microorganisms. you continue with the further treatment. In this, biological oxygen demand was what I wanted to bring in. Quickly, we will move into biotechnology now. Biotechnology is a completely application-based science. Use the knowledge of genetics, use the knowledge of engineering to bring about benefits for humans. That is the sole purpose of biotechnology. Here we need to revise the understanding of genes. We need to revise the understanding of transfer of genes. Let's get back to bacteria. There are three modes, there are three ways of sexual reproduction in bacteria that you have studied. And some understanding of this bacteria is necessary for understanding biotechnology. One, bacteria have two types of DNA. One is the chromosomal DNA and the other is plasmid. Plasmids are considered to be self-replicating, short, circular DNA. Short, circular, extra chromosomal DNA. Self-replicating, that means they can make multiple copies of themselves. Plasmids can make multiple copies of themselves irrespective of the cell dividing. Advantage of this is, again, in let's say in a colony of bacteria, there are a few bacteria which have plasmids for antibiotic resistance. Some of them don't have these plasmids. And remember, not all bacteria need to have plasmids. There can be bacteria without plasmids also. Now, if an antibiotic is put into that medium, the bacteria with this and with the plasmid having the resistance to that antibiotic will not be affected by that antibiotic, whereas the bacteria which do not have this plasmid with the antibiotic will be affected by the antibiotic. In this case, a process called conjugation happens if this bacteria is capable of bringing about conjugation. The plasmids will multiply and the bacteria will transfer these plasmids to those bacteria which do not have the plasmids. Here, what are they doing? It's a survival strategy. Some of them have the advantage of overcoming the threat to survival by these antibiotics. They are transferring it to the ones who don't have it. So without increasing the number of this population, they are able to make the entire population strong. Plasmids are a survival tool for bacteria, and plasmids we exploit in biotechnology. So biotechnology allows us to, earlier when we discussed strategies for enhancement of food production, we were talking about the multiplication of organisms, reproduction complete. If you want a particular characteristic, earlier days, if you want a particular characteristic to be expressed, you want to select the individual organism with that characteristic and make that organism reproduce. But what if I want only a particular characteristic, only a gene product, but not the entire organism? Biotechnology allows us to do that. The technique that is being used is recombinant DNA technique or technology. Recombinant DNA, you have DNA, you know the gene. Gene is a sequence of nucleotides on the DNA. If I know where the gene is on a particular chromosome, if I know what the sequence of nucleotides on that DNA, and if I am able to cut only that segment of the DNA and put it into another DNA, incorporate it into another DNA and multiply the DNA to bring about transcription and subsequently translation, I will be able to bring about the product of this gene without actually bringing about reproduction of the organism. So recombinant DNA is a DNA which has the gene of interest. Let's say there is a host DNA, a DNA into which we will introduce some new gene of interest. I'll take an example. Let's consider insulin. Insulin gene is present in human chromosome. Whatever is the chromosome number, let's not bother about it. The chromosome number and loci is not our interest right now. I cut the insulin gene, take the insulin gene and incorporate it into the plasmid of a bacterium. Now this plasmid can multiply. I make the bacteria which have the plasmid with insulin gene multiply and increase the number of those bacteria. I will stimulate this insulin gene by specific chemicals and make this insulin gene produce the product of itself, that is insulin protein within bacterial cell, and I extract it. So in biotechnology, in the recombinant DNA technology, I take the gene of interest, I incorporate it into a host, into a vector or vehicle. I make the vehicle get into a host which will carry the vehicle. I will stimulate the gene of interest and bring about the product expression, extract the product, use it for human welfare. This is the sequence of events. Some specific details about how it is done, I'll tell you. Step by step we will go. To extract the gene of interest, the sequence of nucleotides on the DNA is known. There is a specific class of nucleases, enzymes which will break down DNA, endonucleases. There are two types of nucleases, exonucleases and endonucleases. Exonucleases breaks DNA from the ends. Endonucleases will break DNA at the centers, somewhere in between. A class of endonucleases is called restriction endonucleases, which will act at particular sequences called palindromic sequences. Palindromic sequences means the sequence which will read the same from both sides. It's like the word Malayalam. You read from left to right or right to left, it reads the same. The spelling is the same. So the sequence where 5' to 3' and 3' to 5' is the same is called palindromic sequence. Restriction endonucleases will cleave the DNA, will cut the DNA at palindromic sequences. You take the DNA sample, treat it with restriction endonuclease. Even before we do this, a lot of other treatments should happen. Let's assume that all of that is done and we have raw DNA available in our hand, in our sample. You treat it with restriction endonucleases, it will cut it at specific locations. For this, the prerequisite is we need to know the sequence of the gene, we need to know what restriction endonucleases are present on either side, we need to know the palindromic sequences on either side of the gene, we need to know what enzyme will act on it, and we need to have those enzymes. Assuming all of that is taken care of, we cut the DNA at these locations and the fragment that we take has the gene of interest. Amplify this gene of interest. Amplify this gene of interest by the technique of PCR, polymerase chain reaction. Polymerase chain reaction is you take this gene of interest in a medium where there is Taq polymerase, Thermophilus aqueous polymerase, which can withstand high temperature. Add some primers, add the nucleotides, synthetic nucleotides into the medium, heat it, the DNA fragments will break, cool it, the polymerase will make the strands. So you are doubling the DNA with every cycle of polymerase chain reaction. So increase the content of DNA to the extent of your requirement. Now second, you have the gene of interest amplified to the extent that you require. You want to transfer this into a vector. Selection of a suitable vector is also important. Plasmids are important vectors for gene transfer. Plasmids will have a region called origin of replication, ORI region. So the action of the restriction endonuclease should not be in the ORI region. If the REN, restriction endonuclease, strikes the origin of replication region, the plasmid cannot replicate. And there will be certain genes present in the plasmid. A good vector should have the origin of replication region intact, that is the REN, restriction endonuclease, should not act on that. It should have a selectable marker. Selectable marker means, let's say the vector has a gene for ampicillin resistance. Now if this ampicillin resistance gene is intact, bacteria with this vector treated with ampicillin will not have any change. If I incorporate my gene of interest into ampicillin gene, the bacteria with this plasmid technically has the ampicillin gene, but practically the gene for ampicillin resistance has been replaced with the gene of interest for us. That is, let's say, insulin. Now if you treat it with insulin, if you treat it with ampicillin, this bacteria will get affected, right? So a good vector will have the origin of replication and a specific marker. Let's assume this is we are taking the example of insulin. We had the gene of interest amplified. The vector also is treated with the same restriction endonuclease that was used to separate the gene of interest, and these two are incorporated. Next, we want these plasmids to be taken up by bacteria in a medium. The plasmids have to be transferred to a large number of bacteria and they have to be multiplied. You stimulate it either by heat or by chemicals or gene gun, biolistics, whatever is the method you stimulate it, you stimulate the bacteria to take up the plasmid. Now there are three possibilities. There is one possibility where the bacteria has no plasmid at all. Second possibility is the bacteria has plasmid, but that plasmid does not have the gene of interest. And the third possibility is the bacteria has plasmid with the gene of interest. We want the third category. You treat this bacteria, this entire medium, this entire colonies with another antibiotic. Let's say there was ampicillin resistance is where we have incorporated. Let's say there is tetracycline resistance also in this plasmid. Bacteria, that is category 2 and 3, both have tetracycline resistance, so they will not respond to tetracycline. But bacteria colony 1 will get destroyed by tetracycline. At tetracycline, the first colony is removed. Those with plasmid do not get affected. Now we have to remove the ones which don't have the gene of interest. How do we do it? We know the gene of interest is incorporated into ampicillin, that is the selectable marker. The one with the bacterial colonies with plasmid having the ampicillin resistance gene intact do not have the gene of interest. Add small doses of ampicillin in a good culture medium, in a nutritive culture medium. The bacteria with plasmid for ampicillin resistance gene intact will grow because it is not affected by the ampicillin present in the medium. The ones with our gene of interest, growth will be suppressed. Remove the ones which are thriving because they don't have the gene of interest as the ampicillin resistance is intact in them, and select the ones which have whose growth was suppressed. Now the ones whose growth was suppressed is indicative of ampicillin gene ampicillin resistance gene being destroyed, where the incorporation of our gene of interest has happened. Select that, culture that in a good nutrient medium, transfer that into a bioreactor, add the nutrients, culture this bacteria to the optimum level, proper conditions, stimulate the expression of ampicillin resistance gene. Now the structural gene has been replaced, but the regulatory gene component for ampicillin resistance is intact. Add ampicillin in low doses into this medium. The number of bacteria is optimum. You are adding ampicillin in lower doses. That means it will not kill the bacteria, but it will stimulate the expression of ampicillin resistance gene. But here the structural gene is not ampicillin resistance, it is of insulin. This bioreactor, the bacteria will start expressing the gene present in between this ampicillin resistance gene, and that gene is insulin. You are expressing the gene of interest. With that, you have the production of insulin, and the removal of this from the medium is called downstreaming process. So the sequence is pretty simple. The sequence is you take the DNA of interest by treating it with restriction endonucleases, amplify it by PCR, polymerase chain reaction, select a suitable vector, treat it with the same restriction endonuclease, bring these two together, form a hybrid DNA, that is the vector, the plasmid with the gene of interest, that is the recombinant DNA. Make the vector get incorporated into a host bacteria, stimulate the bacteria to take up this vector plasmid, select the ones which have taken up the plasmid with the gene of interest, amplify its growth by growing it into a good culture medium, transfer this bacteria with the gene of interest into a bioreactor, stimulate the expression of that gene by addition of the chemicals which will trigger the expression of the gene. When the gene that has to be expressed is stimulated, what is being produced is what we want. Take it out from the reactor by the process of downstreaming, purify it, package it, sell it, use it. This is the entire sequence of recombinant DNA technology and the production of biotechnological products. And the usage and the applications of biotechnology is various. It is used in agriculture. In agriculture, one thing you have to remember is the presence of Bacillus thuringiensis toxin gene in plants, in cotton, Bt gene. This Bacillus thuringiensis gene will produce a protein called cryprotein, crystalline protein, which is toxic for certain insects. So when these insects bite on the cotton plants, the protein goes into their gut and it will break down the walls of the gut, making the insect die. So there is resistance to these insects, natural insecticide. And there is one more natural process that happens in most eukaryotic plants, natural defense mechanism, is RNA interference. In tobacco, the resistance to nematode infestation is brought about by RNA interference. I cannot explain that process right now, but RNA interference, one thing you have to remember is, it is not an induced mechanism, it is a natural defense mechanism in several eukaryotic plants, eukaryotic cells. Then the application of biotechnology in medicine, we took the example of insulin. Biotechnological products are used for gene treatment, gene therapies, where adenosine deaminase deficiency is replaced in overcome by inducing the lymphocytes with adenosine deaminase gene. Gene therapy is also picking up, and over a period of time, there will be possibilities of treating several genetic disorders by gene therapy. One complication of biotechnology for the nations is the issue of biopiracy. You have to be aware of biopiracy. Biopiracy laws needs to be strengthened worldwide. Anything that is native to a particular country, anything that is being produced, biopatency and biopiracy. Biopatency is protecting whatever biological products, whatever biological innovations and inventions that you have made. Biopiracy is using somebody else's invention or using something for your own credit without having done to get that credit, or they have done something to get that credit. So biopiracy and biopatency are issues which are deliberated a lot in the world today. Most of the important concepts of these 4 chapters I have touched upon, and as I always say, this is a short session. I can only refresh your memory on key concepts of the chapters that we will be discussing. With this discussion, you can go back, take out your books, read through the chapters, and you will be able to understand all the concepts better. We'll take up a few questions. Describe gene transfer in bacteria. Jagolu. There are 3 natural ways of gene transfer into bacteria. Conjugation, I told you. If the bacteria has a fertility factor, there will be conjugation tube produced and plasmids will be transferred to bacteria which don't have it. Conjugation. Conjugation is always beneficial because it is transfer of plasmid. Plasmids have advantage. The second one is transformation. Transformation is there is bacteria in the medium and there are stray DNA available in the medium. Some of the DNA can get into the bacterial cell and get incorporated into the bacterial cell. Transformation. Entry of some random DNA into a bacterial cell and getting incorporated into the bacterial genome. Transformation can be beneficial or harmful depending on the nature of that DNA that gets incorporated. The third one is transduction, where bacteriophages will push in their genetic material into the bacteria. Bacteriophages are parasites of bacteria. They will inject the bacteria with their DNA or RNA, whatever is their genetic material, to use the machinery of the bacterial cell and eventually destroy the bacterial cell, and they multiply. So transduction is always harmful. Conjugation is always beneficial. Transformation can be beneficial or harmful depending on the nature of DNA that gets in or the nature of genetic material that gets incorporated. Transduction is always harmful. Selection, somebody has asked, explain about selection, natural selection. Selection is one of the oldest methods of picking up the plants or animals with the desirable characteristics. It's a long drawn process, but a good process to choose the one with the desirable traits. I think any other question that I feel like need to be answered, I will answer in the comment section. For now, I think we can close. These 4 chapters are small chapters. Biotechnology is a very important chapter. Food strategies for enhancement in food production, microbes in human welfare can give you some good questions in the exams. So those small chapters, you have to just get in touch with the concepts there, know the few basic facts given in those chapters. You should be able to do good. Thank you very much.